Okay, ooh, that is super washed out of my computer. All right. Okay, cool. Let me turn off this front light and see if that helps. All right, that's a little better. Okay, all right. So last chapter, right, chapter 6, we were talking about the process of mitosis, which is how well, let me ask you guys, what is the process, like, why do we do mitosis? What's the purpose of mitosis? Healing. Excellent. What else? Cell reproduction. But why do our cells need to reproduce? Yeah. Okay, so yeah, replace cells that are worn out or damaged, right? Yeah, exactly. They get old. We got to get them out of here, replace them. Okay. All right, one more thing. Growing. Yep. So just regular ass development. Okay. And then remind me, how many cells are produced at the end of mitosis? 2. And what is their genetic relationship to each other? They're identical. Okay. So if we only have reproduction through mitosis, we're not going to have genetic variability, which we already decided was a problem. And that's why prokaryotes that can only asexually reproduce have to do this weird horizontal gene transfer nonsense that we talked about last first day of chapter 6. Okay. We're not even going to talk about prokaryotes at all because they don't have sex. So we're only talking about eukaryotes now. Gotta get rid of this. Okay. Okay, think way back to the very beginning of the semester when we were talking about evolution. Why does genetic variability matter? Yeah, exactly. Having that genetic variability allows different traits to be acted upon by natural selection. And if we lose genetic variability, we're more likely to go extinct. Okay. All right. So in eukaryotes, the way that we get genetic variability is through sexual reproduction. There's going to be 3 ways that during this process of meiosis, we are going to see, well, actually, 2 ways during meiosis and 1 outside of it, where we are going to see the sources of genetic variability. So when we get there, I'm going to like make it bigger in your notes and forget about it. Okay. Okay, so we already talked about how mitosis is going to give us genetically identical cells. And this is how we are going to do asexual reproduction. If for asexual reproduction, you only need 1 parent, right? So you're going to have these little flatworms, right? They can do binary fission to just like bloop themselves in half. And now we got 2 little guys that are genetically identical. And there's other ways we don't have to worry about. You don't spend your time learning these 3 different ways. Okay, we're just comparing and contrasting. Okay, so I guess the takeaway is that eukaryotes can do sexual and asexual reproduction. Okay. All right, what do you guys think? What are the pros of asexual reproduction based on what you just learned? Yeah, so you're independent. You ain't need nobody else. I ain't need no man. Yeah. Okay, so a con might be if you have a mutation, especially a harmful one, like that's going to stay in the population until it gets selected out, right? And then you decrease your fitness, and then it's bad for you. Yeah.
Also,
okay. Yep. So if you know you're good, right, and you've got a stable environment, and your genes are like adapted to that environment, you're like, why would I shuffle this deck? I got a winning hand. Like, I'm stay here. Okay, awesome. All right, anything else we want to add? Right, so yeah, so they're genetically identical, so we're going to reduce our genetic variability. Right, yeah, which can have long-term evolutionary problems. Okay. So some organisms, these are the little guys that I studied for my undergrad and graduate research. These are little pemphigus aphids. So inside of these galls, there's these little bugs that trick the plant into making them a bug house. So what these guys do is they have both a asexual and a sexual phase of their life cycle. So they actually can clone themselves. So in the early spring, when the plants are just starting to like poke their leaves out of the buds, the eggs of these aphids will hatch, and the what we call the stem mother is released, and those moms will fight to the death over this favored spot on the leaf where the leaf blade meets the little stemmy part called the petiole, and they will like there's like two of them there, and they're like facing off, and one of them will like stab their leg and rip out the guts of the other one because they want to get that spot. Because what they're going to do is they're going to trick the plant into like they release hormones. The bugs release plant hormones. So crazy. And trick the plants into folding this over. They change the gene expression of the plants to make this little house, and the plant is pumping all the sugar that it makes through photosynthesis into that spot and is feeding the stem mother, who is asexually reproducing. She's cloning herself while she's inside the leaf of that, like in that gall. Depending on the species you're talking about and the location, late summer, so it can be like late July, early August. These break open, all those clones fly out to go live on the roots of a different plant species and feed there over the summer. They mate, the males stay underground. So they'll mate, and then one of the females will fly to the cottonwood trees, lay their eggs, and die, and then the cycle continues. So when the aphids are in these galls, right, they've got this nice stable environment. They don't want to send alarm signals to the plant to say, hey, there's an invader here, right? We want that, like we need to lay low because otherwise the plant's defenses are going to kick us out. So we don't want to have sexual reproduction in that stable environment. We don't want to mess with those genes because they're perfectly suited to do this manipulation. But then but we still need to maintain some of that genetic variability. So there's an option for sexual reproduction at the end of the season. They're so cool. Okay. The number one rule in biology is there's always an exception to the rule. The rule is that all eukaryotes undergo sexual reproduction. So sexual reproduction requires two parents, right? There you need a sperm and an egg. It requires a lot more energy. How many rounds of cell division did we see in mitosis? How many times did we go through the cycle of prophase, metaphase, anaphase, telophase? One time. Meiosis, we're going to do it twice. Okay. And then what we're going to end up with at the end is that our offspring are going to be genetically different than our parents. They're going to have to be similar, but they're going to be different. I don't know if you've ever seen my kids. They all look the damn same as me, but they are allegedly genetically different. Okay, so basically what this is talking about is the Red Queen hypothesis. So when Alice went through the looking glass and she met the Red Queen and she was like, Red Queen's like, we got to run and run, 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 run. And Alice is like, why are we running? We're staying in the same place. And the Red Queen hypothesis is kind of explaining why we see sexual reproduction is because we have this genetic arms race. So if we use the tree and the aphid example, right? The aphids need to be able to manipulate the tree to get them a bug house, right, for their reproduction. But that takes energy away from the tree. So the tree doesn't want that, right? So then their sexual reproduction cycle is going to give them different genetic combinations that's going to make them resistant to the bugs. Well, then the bugs are going to evolve, and we're just going to keep running and running and running and staying in the same place. Like the difference between, right, the manipulator and the manipulee, that's not a word, is the same distance, right? But we're always trying to outdo the other, but we're not really getting anywhere. Okay, so that's an explanation of why we see sexual reproduction. Okay, so let's talk about here's preview of what's coming up next, right? So we're gonna have this process of meiosis, okay? Actually, let me start here, okay? So for sexual reproduction, we're gonna have a quick little look. We're not gonna stress about the details, but we're gonna look at them so that the life cycles so that we can understand what's going on. If you take my Bio 182 class, then you have to learn all the details, but you're not in that class, so we're just gonna look. A little peeky peek, okay? So we're gonna have specialized cells that are gonna form gametes. We call those things sperm and egg. Our gametes are gonna end up being haploid, which is gonna give us our sexual reproduction rate. So we're gonna have egg meet sperm, boom, fertilization happens. Once we have that fertilization, now we're gonna be back to a diploid zygote, okay? The way that we make gametes is this big old crazy long process of meiosis. We've got we're gonna do that prophase, metaphase, anaphase, telophase twice. So we've got meiosis 1 and we've got meiosis 2. All the cool shit happens in meiosis 1. Okay. All right, that's a little roadmap of where we're going. Okay, let's talk about life cycles first. Okay, and this is where I get to turn this. There are some other differences that we'll point out as we get along to them. Okay, so what mitosis is asexual reproduction, genetically the same. Meiosis is sexual reproduction, genetically different. Okay. The way that I taught my daughters to remember this when they were in high school is that mitosis makes more toes. Meiosis is sex. I'm a good mom. Now you will always remember. All right. Yeah, I will draw this on the board. All right, so whenever we're looking at a life cycle, we're gonna build this thing that I call a ploidy fence, okay? And on one side of the fence, we're gonna have a diploid stage of life. Remind me what it means to be diploid. 2 sets of chromosomes, okay? And then what's the other form? Haploid, and 1 set of chromosomes. I call this haploid town over here. All the cells on this side of the fence are going to be haploid, have 1 set of chromosomes. This side I call diploidville. Okay. So, here's you. You are a grown-ass diploid multicellular organism. Woohoo! Depending on what gonads you got randomly selected, everybody, doesn't matter what gonad you have specialized cells in your body that are going to do this process of meiosis. Okay, the only way to get from diploidville to haploid town is through this process of meiosis. So this is our one-way gate in our ploidy fence. Okay. Hey, spoiler. How many cells do you think are going to result from meiosis? Yes, we're going to get 4 cells. Okay. So this one cell at the end, right, we're going to get 4 cells, which is different than what we got in mitosis. Okay. Because this is going from diploid to haploid, right, that's all these gametes that we're going to end up with are going to be haploid. Okay. Now, depending on what gonads you're born with, these are either going to become sperm or eggs. Now we have arrived at our other fence or our other gate in the haploid fence is what I should say. And this is the process of fertilization. Okay. When we have the fusion of egg and sperm, that is going to give us our diploid zygote. Okay. This is the first cell in our diploid stage and it results from fusing of the genes from sperm and egg. This is how we get one set of chromosomes from dad and one set of chromosomes from mom. Okay. And you guys are used to this by now. This arrow is hiding a lot of details. Yay. Mm-hmm. All right. Screen. There we go. Okay. All right, so we've already looked at the definitions of ploidy, right? How many sets of chromosomes do we have? Have we talked about homologous chromosomes? Okay, let's talk about it. Okay, a homologous chromosomes only exist in diploid, or this isn't technically true, but we're not splitting hairs. So we're going to pretend like this statement that I'm going to say is true. Okay. Homologous chromosomes only exist in diploid organisms. A more accurate statement would be homologous chromosomes do not exist in haploid organisms. Because we can be tetraploid or quadruploid or sexaploid or octoploid, we can have all sorts of different ploidy numbers. But for us, because this is Bio 100, like, let's just talk about diploid and haploid and pretend that that's what the only ones that exist. It's complicated enough. Thank you, please. Okay. So homologous chromosomes, or I'm going to start calling them homochromos now because homologous chromosomes is way too many syllables to say. Okay. This is a set of chromosomes that have the set, like they're the same size, they have the same genes, right? So give me a trait. Brown hair. Okay. So from mom, you got the allele, right? The gene version that for brown hair. Do you have to, on the chromosome for dad, do you have to have brown hair? No, you could have blonde hair, right? But you have to have a gene for hair color. If we had a gene for hair color here and hair color here, those are not homologous chromosomes. Okay. So these are like our chromosome number. Chromosome number 1 we got from mom, chromosome number 1 that we got from dad. Chromosome number 2 that we got from mom, chromosome number 2 that we got from dad. Okay, cool. I don't like that my screen keeps washing out. That's so stupid. Okay. Oh, now I lost my clicker. Okay. Otherwise, we're good on this slide, right? Okay. For the sake of time, let's not do it. We're skipping the variations of the life stages, life things. Okay. We'll just use what animals do because we're used to that. Fungi do weird shit where they don't want to be They don't want to be diploid. As soon as fertilization happens, they're like, ew, gross, and they immediately undergo meiosis to get back to being haploid. Plants do whatever they want, where they have multicellular haploid and diploid stages. Plants are cool, but their life cycle is weird. Let's move on. Okay. As we already alluded to, fertilization, the technical definition is the fusion of gametes. Okay, we've got one set of chromies in the sperm, we got one set of chromies from in the egg. When those two cells fuse, we are going to end up with that diploid zygote. Okay, this is our this is our first source of genetic variability. Okay, so write that down big in your notes. And in fact, no, I didn't. Okay. Not the number one, not like ranking them, but the first one that we came across, source of genetic variation, comes from the process of fertilization. That's really important. So that means we got 2 more to go. And that's because and we're going to get there when we talk about what's hiding behind this arrow of meiosis. The genes that get selected to be inside of a sperm cell and an egg cell is kind of random. Okay, so any random sperm that's going to fuse with any random egg, they're going to have different variations of those genes. And we're going to talk about how we make those. Okay, so meiosis is, so here we go. This is all the details. We're not going to talk about it on this slide though. Okay, this is all the stuff that's hiding behind that arrow. This is how we produce gametes. Okay, so if we look back at the drawing on the board, we have this specialized cell in our bodies. It starts out being diploid, and we are going to go through this process of meiosis. During the process of meiosis, we are going to take that diploid cell, boop, boop, make 4 haploid cells. Okay, we are going to split up the genetic material and half it. And those are going to become our gametes. Unless we're talking about plants, and then they do weird shit that we don't have to talk about in this class. All right, that's fine, whatever. Okay, so if we don't have this ability to cut our genetic material in half, over just 10 generations, we would have so many chromosomes that we wouldn't be able to contain them all. So let's have a halving mechanism. Okay. All right, so just some other terms that we need to make sure that we understand, right? So gametes, these are our haploid reproductive cells, sperm and eggs. Somatic cells are all of the other ones. Okay, these are your body cells. So this cell that undergoes meiosis is a germline cell. Okay, but all the other cells in your body are somatic cells. Okay, and then we figured that out using these gross-looking worms. Okay. All right. So again, meiosis is we're going to use the same stages and we're going to call them the same names, but we're going to give it a last name. Brain and heart are somatic cells. Yep. Yep. It's only the cells that are your reproductive cells. Okay. All right. So just kind of a quick overview, right? So we are going to have our specialized cell. We are going to synthesize, make our copies of our DNA. In prophase 1, we're going to do what we do in prophase. We're going to condense up into Cheeto form. We're going to get rid of nuclear envelope. We're going to line those bitches up. We're going to pull them apart. We're going to remake 2 cells, and then we do meiosis 2, where we undo like that nuclear envelope that we just made. We got to undo it. God damn it. Okay. Then we're going to line them up. Line up our chromosomes again, yank them apart, reform our nuclear envelopes. Dun, 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 dun. We have 4 haploid cells at the end of this process. Okay. Okay. All right. Should we get into the deets? All right, how do we feel about this? We know what genes, alleles, chromatin, homochromos. You guys are good on those definitions? Okay. If we get to it later and you're like, wait a second, what the hell are we talking about? Just stop. Okay. Okay. So here we go. We're start with meiosis 1. This is where all the interesting parts of meiosis. Okay. All the things that keep it different than mitosis are going to happen in meiosis 1. So when I say to you, at what stage do the does crossing over occur? You're going to be like, shit, well, I know it's meiosis 1, so the last name of the process is 1, because that's where all the cool shit happens. All right. Let's talk about prophase. Okay, prophase 1. So same things that we're going to see, right? So some similarities as mitosis. We got to get rid of that nuclear envelope. Get out of here. We need to move these chromosomes around. And your nuclear envelope, you're getting in my way. Get the F out of here. Okay. We also got to pack up our DNA. We got to go from spaghetti form to Cheeto form. You don't know what that means. Okay. Where we're going from chromatin, where it's kind of like relaxed, and then we're going to pack it up really tight into chromosomes so we don't lose DNA, that we don't have parts of the DNA breaking off. Okay. Right. So back in S phase, right, that was the DNA was duplicated back there. All right. New things that we only see in meiosis during prophase 1. We are going to see synapsis, where our homochromos synapse together. Okay. So chromosome 1 from dad, chromosome 1 from mom are just going to come and hang out and stand next to each other. And the reason that they need to do this is so that they can swap chunks. Pop off that arm, pop the other one on. Okay. So let's have a closer look at that. Okay, so here's our synapsis. Okay, so this kind of purpley blue is 1 chromosome. The homologous chromosomes shown here in red. And what is happening, right? They're going to come in association. They're going to create these kind of weak bonds with holding these proteins together, right? They're they're buckled in with these little proteins that can unbuckle pretty easily. And what they're going to do is that they are going to just do a little swap. Okay. So here's my my analogy. I got a red marker, got a black marker. During synapsis, those associate with each other. Homochromos synapse together. And then we're going to do crossing over. And we're just going to swap a segment. Okay. So now each of these homologous chromosomes have a unique genetic combination that didn't exist before. Okay. Hey, source of genetic variability number 2. Hell yeah. Okay. In fact, I think I might have that on the next slide. I do. Okay. Crossing over. This is going to be where we see homologous chromosomes exchange material with, importantly, non-sister chromatids. What do you guys think I mean by non-sister chromatids? They're not identical. Okay. So, and we remember that we've got these sister chromatids being held together. These are exactly, allegedly, genetically identical. These are exact copies of each other. So if you have an allele for brown hair, you have an allele for brown hair. Okay. But if we have an allele for blonde hair on our non-sister chromatid, right, they're homologous, They still got hair color, but they don't necessarily have to be the same. Okay. So if I did crossing over with sister chromatids, well, that was a waste of time. I got 2 red markers with red lids. Okay. So, okay. So we're really seeing a mixture um, of the genes that were inherited from what will become the grandparents of a kid that results from this. That was more complicated than it need to be. Okay. The point is, new genetic novel combination didn't exist before. Okay. All right. That was prophase 1. That's cool. Synapses and crossing over. How important. Okay. Now we're going to move on to metaphase 1. Okay. We're still lining shit up along the equatorial plane, but what we're lining up is different. Okay. What we saw in mitosis over here, we saw the sister chromatids line up along that equatorial plane, and the sister chromatids are what was pulled apart, okay, during anaphase. But now, in meiosis 1, we are having the homologous chromosomes line up. Okay, so let me draw a picture of that to make it like hella obvious. Goodbye, animal life cycle. Okay, so what that means is, like last time when you guys were doing mitosis, you guys drew your little X's like this, right? And then your anaphase. You drew something like that, but you guys did a better job than what I just did. Okay. When you draw meiosis 1, you're always going to tell me the last name of the stage that you're working in when you're working in meiosis. Because if you say metaphase, I'm going to assume that you're talking about metaphase of mitosis. If you say metaphase 1, I'll know what you're talking about. If you say metaphase 2, I'll be like, no. Okay. All right. So what's happening in metaphase 1 is we have, let me use different colors. We have a set of chromosomes from one parent lining up next to the homologous chromosome. Okay, so this is chromie number 1. This is both chromies number 2, chromies number 3. Okay, homologous chromosomes. Okay. I'm gonna talk about it right next. Okay, good questions, right? That means you're thinking like in the right direction. Okay. So our spindle fibers now, instead of like right in metaphase mitosis, they attach to the sister chromatids. Now they're like chonking on to the whole damn chromosome. Okay, so we're going to be pulling those homologous chromosomes to the other ends. Okay. Okay, we good? Okay, here we go. Our third and our last source of genetic variation occurs, oops, let me put that back, occurs during metaphase 1. Okay. And this process is called independent assortment. Okay. And what this means is that we're going to see our homologous chromosomes separate into the gametes that we're eventually going to produce independently of each other. Okay. So during this process of how they're lining up along that equatorial plane, right? On this side, I drew the blue one. This side, I drew the green one. Green, blue, green, blue. Like whatever's happening with chromosome number 2, chromosome 1 doesn't care. They're like, you do what you want, bro. I'm gonna do what I want. Leave me alone, okay? So what that means is that every time we have metaphase occurring, because we don't just do meiosis one and done. We're doing this shit all the time. Lady, ovary havers, y'all were born having completed meiosis 1. Okay, so what I'm gonna do, we're gonna try a little spearmint. And we're gonna see the variation of how these homologous chromosomes can line up. Who's the goodest coin flipper in the class? All right. Okay. Do we want green to be heads or tails? Okay, green is heads. You're gonna flip it 3 times. Okay, so go ahead and flip it and then tell me what you got. Tails. Okay, so that means that blue is going on this side. Okay, and green's going on this side. Okay, go ahead and flip again. Tails again. Okay, last one. Tails again. Okay. What if you flipped it again? Would you like, you got 3 tails in a row? It would have to be a heads next time, right? Does it? Okay. Does it have to be heads though? No. Yep. Each time you flip a coin, the result is independent. Okay. Each time these homochromos line up, they're independent of each other. They don't care what the other sets are doing. Okay. We could do this some more and get lots of different variations, but let's not. Okay. I think you guys get the point, right? Okay. All right. So that's our third and last source of genetic variation is independent assortment. Happens in metaphase 1. Okay. This is just all if we had a 3 or an organism with 3, let me say words. If we had an organism that had 3 chromosomes, 2 sets, so total of 6 chromosomes, these are all the different variations of the ways that the chromosomes could line up along the equatorial plane. Okay. The more chromosomes you have, the more potential combinations there are. Okay. So, there we go. Okay. I think I have to stand like right here. Okay. Yeah. Good. Okay. So, so we did prophase 1. Tell me the cool stuff about prophase 1. What's different than mitosis? Synapsis. Okay. And then crossing over. Okay. That gives us, that's boom, genetics, source of genetic variability. Woo woo. Okay. And then metaphase 1. What are we lining up along that equatorial plane? Homochromos. Okay. Not sister chromatids. Okay. In anaphase 1, we're going to pull those homochromos apart. Okay. So here's what's happening in anaphase of mitosis. We see the sister chromatids be pulled apart, but in anaphase 1 of meiosis, our homochromos are getting yoinked. Now, these homologous chromosomes are no longer associating with each other. Dun, da, da, dun, da, 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 dun, da, da. We are now haploid. Okay, that's another cool thing that happens in meiosis 1. We become haploid during anaphase 1. Yeah. Oh, okay. Oh, dang. Okay. Oof. All right, let's see if it's not yelling at me yet, so we better speed up. Okay, and then telophase 1, yawn, this is the boring stage. Okay, so same things we see in mitosis. We're going to relax that chromosomes back into chromatin. We got to reform our nuclear envelope. And then now we're only halfway there, but at the end of meiosis 1, we have 2 daughter cells that are haploid. What is their genetic relationship to each other? Are these daughter cells that we're going to end up with, are they going to be same or different? Different, right? Because we had crossing over, because we had independent assortment. Okay. For us human ovary havers, these daughter cells are going to go back into interphase and they're just going to like wait until puberty happens. Testes havers, y'all are just making gametes all the time. You don't stop for interphase, you just go straight into meiosis 2. Okay. So important at this stage, if we do go into interphase, we're not going to copy the DNA. The whole freaking point of doing this damn process is to half the DNA. If we make more DNA, well, shit. And we just defeated the purpose of doing meiosis. Okay, so between meiosis 1 and meiosis 2, DNA synthesis, no. Okay, you following me? Okay. All right, let's talk about meiosis 2. This is boring, so it goes fast. Okay, so in prophase 2, we do what we do in prophase. We got to get rid of that nuclear envelope. We got to get our spindle fibers, and we got to go from spaghetti into Cheeto form. So basically, we just undid what we did in telophase 1. Control Z that shit. Okay. So this meiosis 2 looks the same as mitosis. Meiosis 2 looks the same as mitosis. I hope I said that right. Okay. Regular ass prophase. Okay. Now in metaphase 2, now we take those sister chromatids, line them up along the equatorial plane. Same thing as mitosis. We gotta make sure, hey, everybody, just y'all buckled in properly? Where's your spindle fibers at? Are they attached to your sister chromatids? Okay, cool, let's go in. Okay, anaphase 2, same as anaphase of mitosis. We pull apart those sister chromatids.
Meiosis doesn't need DNA synthesis.
No, so
Is that what it's
the-
eliminating?
that's-
That DNA synthesis section?
yes, because if we had DNA synthesis happen here, the cells that we would end up with would have 2 sets of chromosomes. We're trying to get from diploid to haploid so that we don't have a giant genome after a few generations. Okay. So that's why we shush DNA synthesis there. Okay. Okay, prophase. Get rid of nuclear envelope, spindle fibers, spaghetti to Cheeto. Line up those chromies in metaphase 2, attach our spindle fibers to our sister chromatids so we can pull those mofos apart. Okay, same thing. And then telophase 2, we are going to reform our nuclear envelopes around each of the new nuclei, and then we also then undergo cytokinesis, and then that gives us 4 cells that are genetically different from each other. I'm allowing processing time. You guys are good? Okay. Okay, write this down big in your notes. This is an important point. When we are all finished with meiosis 1 and meiosis 2, and we've done the whole damn thing, what we are going to end up with is 4 daughter cells. Each of those 4 daughter cells has 1 set of chromosomes. So therefore, we call them haploid. And each of those cells is genetically distinct from each other and is genetically different from the parent cell that we started with. Okay. The DNA, yes, we have different combinations of those genes. Okay, I know it's hard to probably see from back there, but like, right, this guy's different than that guy's different than that. This is, yes, so this genetic variability is what allows evolution to happen. Yeah. Yeah, yes. So if we like go back to the drawing, like the life cycle thing, yeah, so that arrow that's meiosis, there it goes from 1 cell to 4 cells. Okay.
Why was the original?
This, well, these, uh-huh. So,
That DNA, that gone, that DNA.
okay, no, it's not gone. We just put it in different, we split it up and put it into different piles. Okay, so back here in interphase, way before meiosis 1, we're going to synthesize DNA there. We're going to take those single Cheetos and make them double Cheetos. Okay, so we're going to go from, this to that. Okay.
What I'm asking you.
No, let me finish my answer. Okay. Okay. We're going to split those into like, so that's the replicated DNA. Okay. We're lining them up, chunk it out, chunk it out, chunk it out, chunk it out. Okay. At this phase, this is, well, that we've done crossing over and we've done independent assortment, so they're not identical at this phase. But this is where we then split those. This is where we take that double Cheeto and during that anaphase, take it back to single Cheeto. So we actually had 4 copies of alleles, right? Because we had this double Cheeto situation for each homologous chromosome. Okay, and so we're just going to split these 4 sister chromatids that we have and just give each cell 1 at the end, but it's like a new combination because crossing over happened. This is the parent, well, the parent cell, right? yeah, I guess if you want to think about it, no longer exists because it splits and splits and splits, right? It's been split twice to get this. Yes, that original DNA is going to end up in these 4 daughter cells.
That cell dies.
This cell doesn't die. It's going to pinch off. Half of that cell is here, right? And we've generated a new cell. And then this pinches off, right? So it's like, what's a boat, right? So if you replace all the timbers, is it the same boat? So are these the same cells? Because we split it up a bunch of times. Okay. Okay. Why is this here? We already did that. Okay, cool. We already did that. Oh, yeah, that's because we went back. Okay. Okay. All right. So let me go back to summary of meiosis 1. Okay. This is we just did this, so I'm going to go through it pretty fast. Okay. So prophase 1. This is what it looks like under the microscope. There's a drawing of it. This makes it easier to see. We're seeing these homologous chromosomes pairing up. You see those big old red blotches. Now there's 2 red blotches that are hanging out next to each other. Okay. Metaphase 1, homochromos line up and get pulled apart in anaphase 1. And then we have telophase 1, where we make that nuclear envelope relax those babies back down. So and then in prophase 2, we're going to go back from spaghetti back to Cheeto, get rid of the nuclear envelopes, line up the chromosomes, right, and along the equatorial plane so that we can pull apart the sister chromatids during anaphase 2. Then during telophase, we have our 4 new nuclei that's going to reform the nuclear envelope. We're going to relax back into spaghetti form. Good. Okay. All right. I think I can do this fast. Okay, what if we F up? Okay, here's how it's supposed to go. Okay, so we're going to start with our cell. We've got our 2, in this case, we have 2 sets of homologous chromosomes. They're already duplicated in meiosis 1, right? We're going to split up our homochromos. Meiosis 2, we're splitting up our sister chromatids. This is, yay, everything's right, woohoo. Okay, but if we don't attach our spindle fibers to the right places during metaphase, bad things can happen. Okay, so if we have this thing called a non-disjunction event, which is where our chromosomes don't get pulled apart during either metaphase 1 or metaphase 2, we're going to end up with cells that have either too much or too little genetic material. And so if these eggs get fertilized, or if these sperm fertilize an egg, that zygote is going to have an excess amount of chromosomes. Okay, and for animals, that usually results in things going very bad. And there's developmental stuff. Okay, so this is what it looks like if we have a non-disjunction event in meiosis 2, right? So we have our replicated homochromos. They line up, the spindle fibers attached to them properly. Hooray, everything's fine. And then we have a mess up in meiosis 2, where our spindle fibers, instead of attaching to the sister chromatids, they attach to the homochromo. So we're going to get 2 cells that are normal, 1 cell with not enough, and 1 cell with too much genetic material. And if I was mean to you, I'd be like, I would give you this, and I'd be like, this little box here, and I'd be like, did the non-disjunction event occur during meiosis 1 or meiosis 2, and how do you know? What's the subtext there, Libby? If I was mean. Am I mean? Yeah. I'm nice, damn it. Okay. All right. So what this means, the result is, right? If we have a non-disjunction event, that thing do get fertilized, we end up with this thing called a aneuploidy, where we have either too much or not enough genetic material in a cell. Okay. In animals, this is usually results in miscarriage, where that zygote's not viable and it stops the development. There are some cases, such as Down syndrome, where we have a aneuploidy, we have an extra copy of chromosome number 21. Sometimes, like I have a friend who has a kid that is triploid at chromosome number 18. And usually those kids don't live more than 5 years. There's a lot of developmental issues and stuff. But chromosome number 21, usually people with Down syndrome can live fairly normal lives and are like super chill, happy, kind people. They usually have folks with Down syndrome do have a lower life expectancy than somebody without Down syndrome. Okay, this little video for your studying convenience, comparing the two processes of meiosis and mitosis. And then these are just kind of review studies. Like, I think the rest of what's here, yeah. Okay, everything else from here is review. Okay, so that link is there for your convenience. This is comparing the processes of meiosis with mitosis. Okay, so that's a nice little study aid for you. Here's, I listed them out in bullet form, and then a little table of the differences. This is one of my favorite things to do on exams. Ow, damn it, that hint just landed right on my foot. Ow, damn it. Okay. That was a lot. How we feeling? head full again, like we do. Okay, goodbye, video people.